Everybody, I'm sorry. This is now recording. And okay. Yeah, it is recording. So those of you who just pushed the play button, all what I did is that I introduced myself and I'm talking about how I see statistical genetics fitting into plant breeding. So, all right. Okay. So anyway, I was about to say I'm more than like a statistician than a machine learning person, but I am not anti-machine learning. I think that collectively traditional statistics and machine learning can be used in conjunction to solve some of the biggest problems that we face in needing to increase food availability over the next couple of decades and stuff like that. So what we're going to do for the first, hour and a half or so is look at genotype and phenotype models that help ensure investments in high throughput phenotyping technologies are going to lead to meaningful results. Okay. So, and we're going to go over the basics of the statistical models that we use for both genome-wide association studies and genomic selection. Okay, so what can we do with statistical genetics? We can do G to P models. We can associate genotypes to phenotypes, okay? So the basic statistical model is this. Your Y variable is a trait or multiple traits, okay? Then your X variable is one or more genomic markers, okay? And with this basic concept of the statistical model, we can develop models that model the intricate relationship between genotype and phenotype more accurately. And we can come up with more accurate genomic selection models, which you've seen yesterday when Jessica presented yesterday, Professor Rutkowski. So why is this research important? So it enables the dissection of genomic sources for agronomically important traits. And if we do research in these models, we can handle more data situations. So maybe you will have a trait that is not a continuous trait, but, discrete, like, I don't know, number of leaves on the plant, number of stomata on the leaf, right? And if we use an accurate genomic selection model and apply it to the stuff that was taught yesterday and will be taught tomorrow, we can have a really good chance of reducing the length of breeding cycles. Okay, so here is the basic statistical model. That is kind of like the genesis, if you will, of all of these G to P models. Simple linear regression. So what we have is a quantitative X variable and a quantitative Y variable. And we model the linear relationship between X and Y. So our Y value is, so this is the Y value, the ith observation. This is the X value, the ith observation. This is the intercept parameter. This is the slope parameter. And this is our random error term, okay? So beta naught, beta 1 are intercept and slope, respectively. These are unknown constants, these are parameters. And so then what we have is a data set of n observations of X and Y, and we used it to estimate these parameters, least squares estimates, if you want to get more precise. Okay, so any questions? Okay. And I should actually look to see if there are questions. Right. So in order to fit our models, we have to make assumptions, okay? And if we want to get really precise, these are assumptions that we make on the error terms. So epsilon I is NID 0 sigma square E. What does that mean? So it means that our error terms are normally distributed. That's where the N comes in, okay? They are independent of each other. So if I knew the value of epsilon for the 7th observation, it would tell me nothing about the value of epsilon for the remaining observations, and equal variance. So that means that this value, sigma square E, is going to be equal for every single of the n observations. So what can be done if these assumptions are violated? So for those of you who have taken my classes before, and that's only one in this room, okay, you will know that we want to always diagnose our data set to see if our assumptions are met, right? So if these assumptions are violated, we can transform the trait, okay, and then refit the model and see if our assumptions are met. Or we can, implement like a bootstrapping procedure or something like that, that empirically helps us obtain distributions of our test statistics under our null hypotheses. Okay, so that is the basic framework of simple linear regression, okay? And the next step that we're going to take before we start talking about applying this stuff to agronomical data is we're going to talk about how we change things slightly for when our X variables are categorical, okay? So any questions so far? Yes. Okay, so the question is, what do we mean by bootstrapping? Okay, great. So you can think of it like this. Let's say we have 1, 2, 3, 4, 5, 6, 7, 8 people in this room, right? So if I were to randomly select, a sample of 4 of you with replacement, okay, that is a bootstrap sample, okay? So then if we have, say, a data set of n x, y variables, we can take a bootstrap sample of n observations. So we sample with replacement, recalculate, say, a t-test statistic, okay? And then we can take like 1,000 bootstrap samples of n individuals and calculate a t-test statistic each time. And the really cool thing is that if we sample in such a way that we're sampling under a null hypothesis, okay, we'll end up with an empirical null distribution, okay? And empirical, I loosely mean data-driven distribution of our test statistic under null hypothesis. And then we can use that to essentially make inferences. So we can make a decision on whether or not to reject the null hypothesis. Okay, great question. Okay, so there we go. So let's talk really quickly about a factorial experiment, okay? And let's say that we're out in the field. In Illinois, our field is maybe about, what, a mile to the south? So let's say we're doing a plant density experiment, and we got 3 different levels of density, okay? And then let's say that we want to look at fertilizer, all right? So, and we got 3 levels of fertilizer, right? So that means we got 3 levels of factor A, 3 levels of factor B, okay? So that means that there's 9 possible combinations of factors A and B, all right? Now, let's say that we got a total of 27 plants, okay? So we're randomly going to assign these 27 plants to each of the 9 possible combinations. So we might end up having a data set that looks like this. At each of the 9 possible combinations of factor A and factor B, we have 3 replicate plants, okay? So let's say we want to measure yield on all of the 27 plants. Okay, so with a factorial experiment, if we fit a generalization of the simple linear regression model, we might be able to measure a main effect for density. And I'm kind of indicating a main effect of this factor A by seeing how we have uniformly different colors for each of the 3 different levels of factor B, of factor A, okay? And so this will enable us to quantify the main effect of factor A, okay? So we can also quantify the main effect of factor B, and this is denoted by having different colors that are uniform, that are the same for each level of factor B, okay? And also we can estimate an interaction effect, okay? So this means that the effect of yield is different for each possible two-way combination of factors A and factor B, which is denoted by different colors. So we can also quantify two-way interaction effects of factor A and factor B. In a second, I'm going to go to a statistical model that shows us how we can model main effect of factor A, main effect of factor B, two-way interaction effect. But first, are there any questions? Okay, I see a question on the chat really quickly. May you please remind us of how to assess the assumptions to make sure the assumptions are not violated? Sure thing. So to check for normality, what I would do is I would make a QQ plot of your residuals or do a Shapiro-Wilk test. To check for constant variance, I would conduct, for example, a Brown-Forsyth test or look at a residual plot. To test for independence, I would make a plot where your y-axis is the residual versus, say, time series or treatment levels. And that is a very big picture answer. During the break, I'd be happy to talk about this with you a little bit more, okay? Okay, so factorial experiment. Okay, so let's start talking about statistical models. So this concept, main effect of factor A, main effect of factor B, two-way interaction effect, we can encapsulate these concepts into this two-way ANOVA model, okay? So this is a y value of the kth replicate receiving the jth level of factor B and the ith level of factor A, okay? This is our grand mean. So we can imagine that this is the population mean of y, of yield, no matter what level of factor B or level of factor A you're in. This is the main effect of the ith level of factor A. This is the main effect of the jth level of factor B. This is the two-way interaction effect between the jth level of factor B and ith level of factor A. And just like in simple linear regression, we had our error term, right? We also have a similar error term with a similar set of assumptions, okay? So any questions here? Okay, so there's a question of, let's see. Is permutation the same as bootstrapping? No, permutation is sampling without replacement. Bootstrapping is sampling with replacement. And what if we have one quantitative variable and one qualitative variable? Then how you proceed? You would proceed in, well, you can do a thing called analysis of covariance. And basically you have a quantitative variable. That's kind of a nuisance variable as well as a qualitative variable. I'd be happy to talk about that during the break as well. Okay, so here, the word that I did not say, but is written in here is a fixed effect, okay? So really quickly, I'm gonna give an introduction, 40,000 foot view of fixed versus random effects, okay? So your fixed effect, so here, alpha I, beta J, alpha beta IJ are all fixed effects, okay? From a statistical perspective, that means that these are parameters that are going to be estimated, all right? So these are fixed, unknown constants in your population. And from the experimenter's perspective, if your interest and the factors you're testing only apply to the levels that are in your particular experiment, okay, then you want to have your effect as a fixed effect, okay? Does this make sense? Okay, I see people nodding, okay. What is the base to create my any model? Okay. Okay, so there's a question on the chat. What is the base to create any model? If you could please elaborate on that, I would be happy to take a careful look at it over the break. Okay, so let's see. Oh, yes.
So then the alternative to that is a random effect would be if you were looking to make a general model about the behavior of this variable in response to this treatment.
Okay, so the question was, how would this contrast with a random effects model? Okay, so I'll tell you that the next slide we'll look at that in detail, but my answer I just gave to that was a chickening out answer, okay? So what's going to happen with a random effect is you as the experimenter, you will not only care about the particular levels of your factor in your experiment, but you will care about an entire population of levels, okay? And okay, I'm going to spill the beans. So what that means is that instead of your main effects and two-way interaction effects being parameters that are going to be estimated, instead what's going to happen is they're going to be random variables, okay? And they're usually, we assume they follow a normal distribution, and things get a lot of fun. And the way I'm going to elaborate on the answer to that question is move on to the next slide, okay? So here is a two-way ANOVA model with random effects, okay? First of all, let's acknowledge a visual thing, okay? This model looks exactly the same as the model in the previous slide, right? So what's the deal? Why are we dedicating an entire slide to showing this model with random effects? Well, I already answered. What was the name again? Okay, so in answering Adam's question, I already elaborated on the details that make this different, but let's go over this. So yijk, that's your y variable of your case replicate receiving the jth level of factor B and ith level of factor A, right? And okay, this is our grand mean. Okay, now, okay, this is a random mean effect of the ith level of factor A. So to just, drive this point home, on the previous slide, alpha sub I was a parameter, right? It was a fixed constant value, okay? Because it was a fixed effect. Here, we're saying that alpha I is a random variable. It is quite the opposite of a fixed numerical value. It is a random variable, okay? And it's the random main effect of the ith level of factor A. It is normal, independently distributed, population mean 0, population variance sigma square A, okay? Now we're going to also assume that the main effect of the jth level of factor B is random. So that's also a normal random variable. And we're also going to assume that the two-way interaction effect is random, okay? So what we have here then is, oh yeah, and then also we got our good old error term. I'd be remiss if I didn't mention it, okay? So what happens now is that the only, well, how should I say this? Okay, so your parameters now that you're estimating, okay, are your grand mean mu, your variance sigma square A, your variance sigma square B, your variance sigma square AB, and your error variance sigma square E, okay? So that is in detail how your two-way ANOVA model differs between fixed effects and random effects, okay? Questions? Okay, Okay, so I see over here. Whoa. Okay, can you please tell the main difference between fixed and random effects? Okay, I answered that, I hope. When you randomly pull the treatment factors from a pool of treatments and you apply that results to a whole pool of treatments, then it's random effects. You no longer care about the treatments you use. Okay, so when you randomly pull the treatment factors from a pool of treatments, okay, I'm going to paraphrase this question. Let's say we are doing this experiment here, okay? We have 3 levels of factor A, we got 3 levels of factor B. Do we no longer care about our 3 specific treatment levels in our experiment? That is a good question. It depends. And by the way, my answer to a lot of the questions you're going to ask today are it depends, okay? And Anoop has probably heard me saying it depends so much after taking a year of courses with me that he's probably sick of it. So I would say that it's not that you don't care about your particular levels in your experiment, okay? You're just going to go beyond those 3 levels, okay? And so, you know, they are still going to be important because they're going to help you estimate, you know, these variance components over here, these sigma square A, sigma square B, and sigma square AB. So the levels in your experiment, when you are fitting a model with random effects, you know, you do care about the entire population, which, by the way, is this over here, okay? You care more about the population of possible levels of your effects, you know, main effect A, main effect B, two-way interaction effect. But your data are still going to help you with making those inferences. Okay. Random effects contribute to variability Y. Yes. So random effects contribute to the variability Y randomly. Is that correct? So you can think of it like this, okay? Let's look at this equation here. Therefore, the density, the distribution that Y follows, that is going to be a mixture density of 1, 2, 3, 4 random variables. So in that respect, your random effects contribute to the density function of Y. And everybody, I just got really technical with my terminology, but I just want to answer that question. With your example of density in fertilizer, where are the levels? Were the levels predetermined? Sure, we can say that the levels of each factor are predetermined. Is it right to consider if any of them is random? Yeah, so you can consider the levels of your treatment to be randomly selected from your population of levels. Okay, and now I'm getting down into the weeds. So let's go over one variation. How many of you have heard of mixed models before? Sure, all of you. How many of you are comfortable with the concept of mixed models? Okay, right on. Well, okay, so thank you for being honest about this. Now mixed models might sound scary, but it's not, okay? All what a mixed model is you have a fixed effect and a random effect in the same model. That's it, nothing more. So therefore, you know, we just went over a fixed effects model. We went over a random effects model. You are now qualified to go over what a mixed model is, okay? So YIJK is similar to as it has been defined previously, right? We have our grand mean. And I arbitrarily said that, you know, let's have the main effect of ith level factor A. Let's say me as a researcher, I really care about these 3 particular levels of density, right? So I'm going to treat it as a fixed effect. So then, let's say that, let's see, we had fertilizer. Let's say that, I don't know, we went over to, let's see, here are at agronomy grocery store is Blaine's farm and fleet. So let's say I drove up that way to Blaine's farm and fleet, and I had an entire aisle in the shelf of like possible fertilizers. Okay. And so I just randomly picked 3 brands because I'm a statistician. I don't know what's a good fertilizer. Okay. So therefore, I want to make inferences on all possible kinds of fertilizers. So I'm going to treat this as a random effect. So, okay, so let me ask you all a question that I'll tell you the answer to in a second. A is fixed, B is random, right? What do you think the interaction term is going to be, fixed or random? Random, right on, awesome. So here's, and I got people saying on the chat, random. So here's the deal. Here's a foolproof way to always keep this straight, okay? Random wins, okay? So if you have a fixed main effect and a random main effect, their interaction is going to be random, okay? So in this situation, alpha, beta, ij is a random two-way interaction effect. And then we have our error term. Okay, so mixed models, I went over them because they are so critical for the genome-wide association studies and to maybe a lesser extent, genomic selection models, okay? Mixed models are what enables us to make inferences to help us find genomic regions that are likely to be associated with our trait and to help us make accurate predictions in genomic selection. So with that in mind, we're now going to shift gears in a second and start talking about multi-location experiments, our first application of this stuff. But does anybody have any questions? Okay. So, all right, I'm going to do something cheesy. I'm not going to force you all to ask questions right now, but here, I'm going to give Anoop this microphone. And when like somebody has a question, we just turn it on and give it to people. And I think that's going to really benefit the people who are listening online so that, they can hear us. But I will still repeat questions. I always love repeating the question to make sure I got it. Okay, so are you sure there are no questions? Oh, okay, so I got 2 questions right now on the chat. I will answer them really quickly and then I will move on. Okay, how many random effects can you have in your mixed models? That is a great question. So if you want to use traditional model fitting approaches, I would say you need to make sure that the number of random effects is less than or equal to your sample size. If your number of random effects exceed the sample size, then you need to start implementing some of those penalized approaches that we use for genomic selection, or maybe the Bayesian approaches. And what am I talking about? We will cross that bridge later on today. And then the second question is, what number of treatments would make your treatment a random effect? Ooh, that is a great question. So what is a minimum number of random, what is the minimum number of levels in your factor that you need to get a good random effect estimate? So again, it depends, right? But one time I remember when I was actually doing my defense for my master's degree, somebody like one of the members of my committee said, Alex, you need to have at least 10 observations. You need to have at least 10 levels of your experiments in order to have a random effect. So sure, maybe something like 10, but in the realities of actually fitting the models, I've seen smaller numbers than 10. So, but what you want to strive for, surely, when you have a random effect, you want to have, the more levels, the better. That's what you should strive for. What makes the interaction to be random and not mixed? So that's a great question. it is the underlying statistical theory, really. And honestly, I would probably have to consult one of my textbooks from a graduate school to give a mathematical answer. But just remember, if you have a fixed main effect for factor A, random main effect for factor B, random wins. Now, okay, and I'll answer one more question. When you are fitting a model, how can you decide if some of the variables should be modeled as fixed or random effects? Okay, so when should you decide something should be fixed versus random? It is you as the experimenter who makes that decision, okay? And you use your subject matter knowledge to make the best decision possible. So it's not a statistical answer to decide fixed versus random, but it's more an experimental, practical, within the context of your expertise kind of question, okay? All right, so let's say that we have multiple location experiments, and I'm sure that many of you, or maybe most, maybe all of you, your experimental data are combined across multiple field seasons, multiple environments, right? So let's consider that scenario, okay? Let's say we have seeds, maybe they are obtained from a germplasm bank, okay? And then we end up with, I don't know, let's say a diversity panel, a diversity panel planted with multiple reps in multiple environments, okay? So therefore you have the same genotypes that are measured across multiple reps, multiple environments. So how can we summarize this information? Sorry, okay. My laptop was getting loud and I was making sure everything was okay. So we can tweak the statistical models that we've learned so far to account for genetic, environmental, and genetic by environmental sources. So instead of talking like factor A, density, factor B, fertilizer, let's now talk about genetic, environmental, and G by E. Okay, and then the output from this model are going to be blups and blues, okay, of trait values for each of our taxa. And what are blups and blues? Stay tuned. I'm going to teach you what they are. All right. So here's the statistical model used to obtain blups. Okay. And we've seen a lot of this already. So we have our phenotype of our ith individual. And then we have, you know, our genotype effect is a random effect. Our environment effect is a random effect. Our G by E term is a random effect, and then our grand mean and our error term, okay? So imagine if we fit this model to our multi-environment data of, you know, genotypes. So the output that we'll get from this is a blup, okay? Best linear unbiased predictions of our genotype effect, okay? So let me back up very slightly, okay? When we fit our, you know, this random effects model, we can obtain estimates of each of our genotype levels, okay? So, and I'm using that term estimates loosely because when we have random effects, we are not allowed to call them estimates. We have to call them predictions, okay? So, if we want to obtain our genotype effect for each of our n individuals, of each of our n genotypes, we'll fit this model and obtain a, and we obtain our bluffs. Oh, and sorry, there was a request. Okay, somebody needs to mute the microphone. Okay, and so then also an added benefit is this, okay? We can get our variance component estimates. So remember that our G main effect, our E main effect, our G by E interaction effect, those are all random variables. Those are all random effects. So we can obtain estimates of the variance of G, the variance of E, and the variance of G by E, okay? So that is going to give us variance component estimates, okay? And we can use those variance component estimates to estimate heritability, which you all learned yesterday is the proportion of total variability in our traits denoted here as Y that is attributable to genetic effects, okay? Okay, so another widely used model is a slight variation of what we've seen previously, okay? So the difference here is that our genotype is a fixed effect, okay? And then the convention in the field is to have our environment as a random effect, and our G by E term is a random effect. Then we have our random error term and our random mean. And so then our output is going to be best linear unbiased estimates of the genotype effect. Okay, so and since G of I is a fixed effect, we are allowed to call them estimators. So therefore, we call them best linear unbiased estimators or BLUs. Okay, so let's see. I am about to go over BLUPs versus BLUs really quickly, but does anybody have any questions? Yes, Gina. You don't have to.
I find it obviously your reputation is more bold, but it's going to block the location, block the computer. You don't have any kind of stories about how to go. Once obviously I know people in the model, but at what threshold is it dropping that very important?
Yeah. Okay, that is a great question. I'm going to repeat it just to wrap my head around it. No, it's totally cool. So Gina is fitting a model with a whole bunch of X variables, a whole bunch of bells and whistles, accounting for block effects, interaction effects, the whole 9 yards. So the question is, what should be the decision-making process of when to remove some of the X variables that are unnecessary? Okay, so I actually, I have an example where we go over this. So there's a lot of different strategies. What I've done in the past was I would use a backwards elimination procedure. So to go into the weeds a little bit, there's an optimization criterion you can calculate. The one that I like is Bayesian information criterion. And that will basically tell you, is it worth it to have an additional X variable in your model? Okay. So what we've done in the past is we would start off with the most complicated model we could possibly use, and I'll show it to you in a second. Okay. And then we would systematically remove X variables, calculate our BIC criterion, and then we landed on using the final model that maximized the BIC. So you can do a statistical optimality criterion like that. Okay, I got 2 questions here. Is it possible to get the variance component of the genotype when we treat variance as a fixed effect? That is a great question. So no, it is not possible to get the variance component of the genotype when you treat genotype as a fixed effect. So what I've done in the past when I've had to obtain BLUs instead of BLUPs is I would obtain BLUs, and then I would go ahead and refit the model with G of I as a random effect as presented in the previous slide. And I would go ahead and obtain the variance component estimates there and calculate heritability. So I'll answer one more. Okay. Since all the factors of the model and the genotype are random. Okay, so in the previous page, all of the models, sorry, the genotype effect was random. E was random, G by E was random. That is technically a random effects model. Okay, so okay, let's compare and contrast the fixed BLUPs versus BLUs. So BLUPs, the advantage is, in my opinion, it makes more sense from a biological perspective, okay? if we have a data set of n genotypes, right? That's not the entire species, right? I want to make inferences on the entire species or maybe a subpopulation, right? Or maybe my breeding program. Well, I don't have a breeding program, but I think the BLUPs make more sense from a biological perspective. A major disadvantage and a deal breaker for me sometimes is that a statistical property of BLUPs is that it shrinks your values towards the mean, okay? So if you have a trait with not a lot of variability, you might get BLUPs that are all 0, okay? And you can't do anything with that. If all the BLUPs are the same value, you cannot make any inferences, right? Yes.
Does that mean an advantage when you have highly imbalanced data?
So by
Shrinking.
Yeah.
Be an advantage if you have unbalanced data.
Oh.
You have 2 reps in one environment and you have 5 reps in another one.
right.
These blocks are sort of something that towards the mean, so you would use it.
So the question is, if you have unbalanced data, so you have more reps in one environment than the other, will this shrinkage actually be an advantage? I need to think about this. Off the top of my head, I'm going to say no. I don't think it's going to be an advantage because there's a concept of biasness, right? So you want the expected value of your estimate to be the parameter value, okay? So I would say that under shrinkage, you're going to systematically underestimate the value of that parameter. And I think even with unbalanced data, I would say that that's still going to be a serious disadvantage. But let me think about this. Yes.
And just to go back to what you said for traits with low variability, so 0.
Yeah, BLUPs could be 0 for traits with low variability. So an advantage of BLUs is that they do not shrink the values towards the mean. They are less computationally intensive. Oh, that's disadvantage too. So fitting a random effects model can open up a can of worms in terms of, taking computational time to fit the model. BLUs do not have that problem because you can use traditional, least squares approaches to obtain BLUs. And then another disadvantage of BLUs is that it makes less sense from a biological perspective. Okay. Okay, and then some technical notes. In plant breeding, the estimate of the grand mean. Okay, yeah. Here's the thing that confused me when I first got into quantitative genetics and plant breeding. Okay, in practice, what is done is this. Okay, you fit the models that I've shown in the previous page, and you obtain your BLUPs if genotype is a random effect, and your BLUs if genotype is a fixed effect. Okay. And then in order to convert those BLUPs and BLUs into similar units of measurement and similar range of values of the original experiment, you add the estimate of the grand mean to the BLUPs and the BLUs, and you call the end product of that. You still call it BLUPs or BLUs. And that confused me a little bit, but I just want to give a heads up that that's kind of the state of the art in terminology. And the rationale is that the resulting BLUs and BLUPs after adding the grand mean is going to be the same units of measurement, okay? So also you want to consider transforming your phenotypic data because this will help, meet your model assumptions. Okay, so here's some software that I've used in the past or, graduate students and postdocs in my lab have used in the past to obtain BLUPs or BLUs. So SAS, the advantage is that it's relatively simple to use, okay? A major disadvantage, okay? I'm a firm believer that you don't have to pay several $100s a year in order to use the most appropriate statistical analysis, okay? I think that for proper dissemination of these approaches to the entire world, you don't have to pay any money for it. So that is a disadvantage for me, for SAS. You don't have, I mean, you have to pay an annual license fee. Also, relatively speaking, proc mixed and SAS can take a very long time to compute, okay? So the model that I'm about to show you in a couple of slides, when we tried fitting in proc mixed and SAS like 11 years ago, it took like, it was like taking a day and then we just gave up. An advantage of ASRML is that it can fit very complicated mixed models quickly, okay? A disadvantage, there's a steep learning curve with like the coding, and I still don't get it correct today, even after it's you know, it's been converted into R. Also, there's a really expensive annual license fee. And actually on my to-do list for this weekend, I need to update my lab's ASRML license. R, the advantage is that it's free, okay? However, a disadvantage is that, you know, our programs may not necessarily be as extensively quality control tested as like SAS or ASRML. So you definitely want to double check, you know, your models that you fit in R with a different software, or just do validation. And I see there's a question on the chat, so let me go there really quickly. Where am I? Okay. In a random effect, we estimate one component, which is variance. For fixed effect, we estimate many parameters based on the number of levels of fixed effect. How come random effect is computationally intensive? Okay. Models that fit random effects, okay. The model fitting procedure is an iterative procedure. So you have to make several guesses on the estimates of your parameters, and your guess gets better and better and better and better. And that takes a long time. When you have a fixed effect, you just have to do some simple algebra that a computer can do in a very short amount of time. Why is blue less biologically interpretable? In my opinion, so for example, if you have a data set of n genotypes, okay, you're treating it as a fixed effect, your n genotypes are the only thing you're making an inference for. And I would argue that I would argue that you want to make inferences for an entire species or for an entire population. And that's why, in my opinion, blues make a bluff, you know, having your genotype as a random effect makes more sense. Okay, so here's this model that I was alluding to when answering Gina's question. So here we had a really complicated model. We were looking at a nested association mapping population in maize. So like 26 different biparental crosses, each with one common parent, okay? We were looking at kernel color, okay? And you know, kernel color in maize, like the oranger the kernel color, the more, you know, provitamin A carotenoids there are. So we had two environments grown at Purdue University. We had an incomplete block alpha lattice design, okay? And then our phenotype, okay, this is actually really cool. We had a 1 to 12 scale on orangeness from light yellow to dark yellow. And we included, you know, the whole 9 yards in this statistical model. We also considered, you know, this AR1 by AR1 correlation structure to account for the facts that plots that are closer to each other might be more strongly correlated than plots further apart. And we did this backwards elimination perspective test using Bayesian information criterion to filter out non-significant X variables in our model. We did this in ASRML. Okay, and so that was a punchline of that story. Okay. Could I elaborate on spatial modeling to get row and column effect, if possible? Thanks. Yes, so really quickly, so we had an AR1 by AR1 correlation structure. So you can, so let's say we have a field, okay? and we have rows, okay? So rows that are closer to each other were modeled to be more strongly correlated with each other than rows that are further apart. So that's one part of the AR1. And then columns that are more closely correlated with each other, or ranges that were closer to each other were more strongly correlated with each other as well. And that was the basic idea. And that transforms into an AR1 by AR1 correlation structure. Yes.
If to pose your term as like, for example, I have three like geographic locations. 2 years. Is it better to just make them like 6 environments in terms of like year 1 this location, year 2 this location, like that way, or is it better to just like have a year term and a location term?
Yeah. So if you have multiple environments and multiple years, is it better to have a separate year and environment and year by environment interaction term versus just combine them all into?
Excluding them.
Yeah. So honestly, what I would do is I would make a decision on a data set by data set basis. So the first thing I would do is I would maybe make some visual tools, some like visual assessments to see if like you can see like a difference in year effect and difference environment effect. And if so, then I would have distinctive year and environment and interaction effects. And then if not, I would just have maybe, let's call them, I don't know, location. I would just have a location effect in your model.
Should you like essentially just fit them both and then like
Yeah.
and then that works too.
Yeah,
Okay.
yeah. I think you can also just go ahead and fit model 1 with just a year environment effect versus a year with another model where you just have all locations in as a main effect and see which one does a better job for sure. Yes. Okay, so now let's talk about genome-wide association studies. And remember, when we're doing a GWAS, we're essentially fitting a fancier version of a simple linear regression model with some more bells and whistles. So the purpose of a GWAS is to identify genomic regions associated with the phenotype, okay? So we fit a statistical model at each SNP in the genome, right? So each marker in the genome. So in this picture, we have chromosome 5 on maize, okay? Our x-axis is our physical base pair position on chromosome 5. And then our y-axis is our negative log base 10 p-value, okay? So the taller the lines are, the stronger the statistical evidence is for an association between that marker and the trait under study, which alpha tocopherol, vitamin E levels in maize grain. So at each SNP in the genome, we fit a statistical model to test for the null hypothesis that there's no association between your SNP and your genotype. So in this particular picture, let's see, our peak association is highlighted in red, and we lucked out. Our peak associated SNP is within a gene that makes biological sense, okay? So if we want to talk about this from a marker-assisted selection perspective, markers that exhibit peak association with your traits are potential targets for marker-assisted selection, okay? Okay, so things get a little complicated with the GWAS, okay? And that's because GWAS is often performed on a diversity panel, okay? Diversity panels are collections of germplasm from, that potentially represent your entire species, right? And lots of species have subpopulation structure, right? So here is a diversity panel, the Ames panel of 2,815 maize plants, okay? And then on our x-axis, we have principal coordinate 1. Our y-axis is principal coordinate 2. We can essentially think of these as something similar to principal components of a principal component analysis of our DNA markers, okay? And so our x-axis is kind of collecting our main genetic distinctions between our individuals, and then our y-axis is depicting our second most important genetic distinction between individuals, okay? So what we're seeing here, Dr. Romay et al. Published this, we are seeing different subpopulations clustering into different groups, and these are denoted by different colors, okay? So if we don't account for this population structure in our statistical model, then what we're going to see in a GWAS is our most statistically significant marker trait associations will be due to subpopulation structure, and we don't care about that. We want to find the genes, right? We want to see, what are the genes controlling the trait, and then another source of false positives is familial relatedness. And you can think of this as refined subpopulation structure. And the best explanation of familial relatedness I've ever gotten was, let's say that, you sequence my DNA, okay? And I have a missing genotype at a particular SNP, okay? Then what familial relatedness would say is that if you were to sequence the DNA of my sister, okay, versus the DNA of some random person walking down that sidewalk over there, the observed genotype at the SNP of my sister is going to be a better guess of what genotype is present at my missing marker than a random person walking down the sidewalk. Okay, so we need to account for population structure and relatedness in a GWAS model. Okay, and my opinion is that the good old unified mixed linear model, I think that this is the most effective way to account for population structure and familial relatedness. So our Y variable is our phenotype of our ith individual. And then at each of our markers, at each of our SNPs across the genome, we're going to fit a separate model where that particular SNP is fitted as a fixed effect, okay? And we are looking at estimating an additive association usually between our marker and our trait. And then we have, fixed effects here for this particular data set I used to analyze this. We have 3 principal components of our genome-wide markers to account for population structure, okay? And then we also include our individuals, our genotypes, our lines as a random effect to account for a familial relatedness, okay? And then we have a kinship matrix, or as you were learning yesterday, additive genetic relatedness matrix that is used to, model the variance covariance between our individuals, and hence account for familial relatedness. And then of course we have our error term, okay? Okay, so really quick question. On slide 19, if the information of PC, observed SNP alleles, and line are all from the genome marker data, won't this cause multicollinearity problem in the regression analysis? Fantastic question. So translating this question to quantitative genetics, what can happen often, a disadvantage of the unified mixed linear model is that if you have a bunch of genes that are highly correlated with population structure, then it's going to be then you're not really going to find that association. And so the person who asked this question point well taken. Multi-collinearity can be an issue with the unified mixed linear model, and it's the thing you have to address on a trait by trait basis. So the thing that makes fitting this model a pain sometimes is an issue we've already touched upon, okay? And this is variance component estimation, okay? It's computationally intensive. So there are computational approaches available to reduce this computational burden. And I'm just going to go over them really quickly, okay? So two of them are in the software GAPIT, and then the third one is from a software called Gemma, okay? So the first thing is a compressed mixed linear model, okay? So if we have all n individuals as a random effect, all n genotypes, and then we have an n by n kinship matrix, the linear algebra operations that you're working with involve, n-dimensional vectors, n by n matrices. So what you can do with the compressed mixed linear model is perform hierarchical clustering, okay? And then subdivide your n individuals into G groups, where G is less than n, and hence you're working with smaller matrices, smaller vectors, so it reduces computational time that way, okay? Another approach is population parameters previously determined, or something very similar, MLX. And so what you do here is you estimate your variance components prior to running GWAS. So essentially you'll fit this model without this alpha xi term, okay? You'll estimate your random effects for your groups and your random effects for error, and then you conduct your GWAS using the variance component estimates, okay? And this really does a great job of reducing computational time, because instead of, estimating these variance components, say like 100,000 times, if you have a data set of 100,000 markers, you're doing it only once. However, this is an approximation, okay? It is not the statistically optimal estimate, and it has a tendency to underestimate most significant associations. So a thing that takes the P3D concept that we looked at in the previous slide, but does it, bigger and better, it does a better job of it, is Gemma. So basically, what's happening behind the scenes is that you are replacing computationally intensive technical calculations with simpler calculations. And it enables you to have a statistically optimal variance component estimate at each marker. Okay. And so this results in a similar reduction in computational time as P3D, but it gives you an exact statistically optimal estimate of your marker trait associations. Okay, so great. So are there any questions? I think what I want to do, oh yes, go ahead, please. Right. Okay. So let me repeat the question. So you're saying, so over the past few slides, we went over the MLM plus some modifications of the MLM. And so your question is, what should we trust? Or
Yes.
okay. So yeah, so let's say, okay, if like someone said, Alex, go over to your office and run a GWAS right now. What I would probably do is, I love GAPIT. I co-developed it. I would probably go into Gemma, and I would run a GWAS using Gemma. Okay, and let's say I have a peak association. Okay, and I want to validate it and make sure it is correct, right? So would I go back to the original unified mixed linear model with none of the computational tricks, just to make sure everything is working? I would say Okay, I'm going to be honest with you. I think that's a good idea. With like Gemma, it is giving you the correct statistically optimal solutions, okay? So my expectation is that Gemma is going to give me the same p-value, maybe off once you look at like 10 decimal places because of differences in programming, but I would expect to get the same answer in the original MLM versus Gemma. But I think you bring up a good point. It's always a great idea to think about ways that you can validate everything you're doing and to double check that there's no bugs in the software you're using, or yeah, so always think about ways to like double check your results. Adam?
So if you're running your GWAS and you have multiple locations in multiple years, but you're only putting one value into the phenotype, where does that come from?
So if you have multiple environments, multiple years, what like phenotype do you put in? Or so that's why we calculate blues and blubs.
Okay.
So you would fit those like mixed models and obtain your blubs or blues, pick your favorite one. And then those blues or blubs will be your Y variable for the unified mix on your model and this genomic selection.
Great, thank you.
Yeah, my pleasure. Okay, so, all right, so let's go over genomic selection, okay? You went over this yesterday, but really quickly, just to think about it, we want to predict phenotypic value, well, actually breeding values, right? So that's a typo I need to correct, right? We want to predict breeding values using markers distributed throughout the genome, right? So we have breeding material that are both genotyped and phenotyped, right? So, oh, wait a minute. Well, I'm going here, sorry. We have a training population that are both genotyped and phenotyped. And so we fit our genomic selection model, right? And our Y variables are trait of interest, and our X variables are genome-wide set of markers, right? So we fit this model in our training set. Then we go to some breeding material that are genotyped with the same markers, and we basically plug and chug these genotypic values into our model. We calculate predicted breeding values, genomic estimated breeding values. Then we use this to make selections. So GS is important because it enables selection without having to phenotype individuals, okay? Therefore, you don't have to wait, one, maybe more field seasons to obtain your phenotype, so it can speed up your breeding cycle. So various frequentist and Bayesian models are commonly used in genomic selection, and most produce approximately the same prediction accuracies. So the basic statistical model is that all markers are the explanatory variables, okay? And so we have our Y variables, our trait of interest, and then we have a genome-wide set of P markers, and the number of markers typically exceeds the sample size, okay? So we got an N smaller than P or a P greater than N problem. So why is this an issue? Well, using traditional statistical approaches like maximum likelihood estimation, you cannot obtain unique estimates of your marker effects. So your first solution is to add a penalty that restricts the range of possible estimated values or predictions your markers can equal, okay? And then your second solution is the Bayesian approach, where you can assign a prior distribution on your marker effects, okay? And this is a little bit more technical than I want to have the level of this workshop at, but that's how we deal with the number of markers exceeding the sample size issue. Okay, so one of the most commonly used genomic selection models, which you've seen already, is the RR blood model. So this is our phenotype of our ith individual. So we include all P markers, okay, in a data set as our random effects, okay? Then we have our error term and our intercept, and then the blubs of these marker effects are subjected to the ridge penalty. And what this means is that instead of, equaling any possible value from negative infinity to positive infinity. The ridge penalty is just saying, a finite boundary of possible values that your flux of your marker effects can equal. Okay, so all marker effects are equally penalized so that they are shrunk to zero. And full disclosure, there are 4 people from Colorado State. My postdoc, Dr. Brian Rice, is in your lab, right? And Brian wrote this slide, and I'm going to show some of his work from his master's thesis at the end of the day. And so stay tuned. Okay, so how many of you have heard of k-fold cross-validation before? Okay, 1 person. Okay, 2. Awesome. Well, I'm going to show you what it is, okay? So let's say we want to figure out how good of a job does a genomic selection do at predicting breeding values, okay? And let's say we have only 1 data set, okay? So cross-validation, we're going to partition our data set into k-folds. So we have a picture here of 5 different squares. So this means we are partitioning our data into 5 approximately equally sized subgroups, okay? Now, here k is equal to 5, right? So we're going to fit our GS model on all observations in the 4 blue squares, okay? And then we're going to see how good of a job this genomic selection model can predict our trait values in our test set, okay? And so the way we test how good of a job our fitted genomic selection model is predicting our trait breeding values is that we'll measure the Pearson correlation coefficient between our observed and predicted trait values. Okay, so we're going to repeat this so that each fold gets a chance to be a red square, that is a test set, okay? Then the prediction accuracy, that's going to be our average correlation between our observed and predicted trait values across our folds, okay? So when I show genomic selection results, okay, I'm going to show them with respect to cross-validation. And I think when Professor Diego Harkin talks at 1 p.m., he might show more elaborate cross-validation procedures for G/E-based genomic selection. Okay, so great. So the last part of this first set of slides before we take a break is just to give a hopefully not too painful introduction to linear algebra, okay? And also let me kind of come clean. So I was thinking that we would end this by 10:30 and then we take a half-hour break. But what my gut instinct is that we'll just go through these slides and then we'll take a break. It might start after 10:30, but then we'll reconvene at 11. Okay, so if I'm acting like I'm a little rushed, that's why. But I think we're going to be fine. Okay, let me ask you all a question. How many of you are scared of linear algebra? It's okay to say yes. Okay, cool. I'll tell you, I took linear algebra when I was an undergrad. It scared me so much, I think I had to take the class twice. So I'm going to show you how it works, okay? And the thing that was so enlightening was that I realized you don't have to go to a sheet of paper and like write out all of the complicated math behind the scenes because you can let software do it for you, okay? So once I learned that, I was much less scared of this. So linear algebra is so awesome because instead of doing a whole bunch of calculations like n different times, you can just put everything into a vector or a matrix and you can just do, those calculations just once, okay? So the basic, building blocks of linear algebra are vectors and matrices. So a vector is just, a special matrix with just one column or just one row. So here's an example of X, and Y. Both of these are two-dimensional vectors. And then A and B, these are 2x2 matrices. So each of these have 2 rows and 2 columns, okay? And just really quickly, I also provide some sample screenshots of R code to show you how to draw matrices, okay? So this is how you do it in R. And I think on your own time, you have access to these slides, and so you can study these in detail and see how it works. Okay, so then the next building block is transposing the vectors and matrices, okay? And all what we're doing is we're rotating these 90 degrees. So you just turn the rows into columns, okay? So here's X, which is a vector, 1 and 1. If we want to take the transpose, we're just turning it on its side. We're turning the rows into columns. And then you have the prime subscript to denote a transpose. So here's X transpose, here's Y transpose. Then I'm going to highlight matrix A. So we're turning the rows into columns. So the first row of A is 1, -1. The first column of A prime is 1, -1. And then the second row is 3, 2. Second column is 3, 2. So this is how you transpose our vectors and matrices in R. Okay. So now let's talk about addition. Okay. So if you have 2 matrices that have the same number of rows and columns, oops, and we see here that A and B both have 2 rows and 2 columns, then all we got to do to add them together is just add each of the individual elements together, okay? So that's how we add A and B, okay? And then the last basic thing, before we get into more complicated stuff, is matrix multiplication. So what you need to have for matrix multiplication is that the number of rows of matrix A need to equal the number of columns in matrix B, okay? Then if that is true, then you can basically do matrix multiplication. And I'm going to attempt to talk through what's happening for one of these operations, but let's see. We got 1 times 1, or sorry, so this over here, which is A times B, okay, we're taking 1 times 1 plus -1 times 1. We add them together and we get 0. And if you make a bunch of similar operations, okay, for the remaining elements, you get A times B. And that sounded really complicated, but this is all what we're doing. This is how you do matrix multiplication in R. Okay, so now we're going to start to get to some fun stuff, okay? First thing is the identity matrix, okay? So if our goal is to create a generalizable version of equations for linear algebra representations of GWAS and genomic selection. Oh, I see somebody is writing on this. Okay, so the identity matrix is basically a linear algebra representation of 1, okay? So just like if you take a number and you multiply it by 1, you would get the same number. It's the same idea. If you take a matrix and multiply it by the identity matrix, you should get the same matrix. But so identity matrices are square, which means that the number of rows are equal to the number of columns. And let's see. And then the diagonal element is 1 and the off-diagonal elements are 0. Okay, so this is how you create the identity matrix in R. Okay, and then let's see. Then inversion. This is important because it is the reciprocal. Okay, so in nonlinear algebra, reciprocals are important. So the linear algebra equivalent is the inverse, okay? And a to the negative first power, that is an inverse if a times a inverse is equal to I, okay? And this helps us conduct important algebraic operations. So this is how you conduct an inverse. This is the solve function. Okay. And okay, so now we're going to take some GWAS and genomic selection models, okay? We're just going to show linear algebra representations of the same models that we've seen already. So this is a unified mixed linear model. I'm going over all the bells and whistles quickly because we've seen this already, okay? So at each marker, we use our genotypic and phenotypic data to estimate these parameters here, right? And behind the scenes, linear algebra is used to provide these estimates. So the unified mixed linear model, as seen on the previous page, can be represented in algebraic terms as follows. So we have, instead of the phenotype of the ith individual, we have a vector of n phenotypes, okay? Then we have the grand mean, and then this one is just a vector of ones. This is a vector of our fixed effects, so beta 1, beta 2, beta 3. Those are the slope estimates of the principal components accounting for population structure. This is a vector of our marker effects. And then these are our n different lines accounted for as random effects. Then our error terms. And then if we want to fit, our unified mixed linear model using linear algebra, we have this monstrosity of linear algebra operations, but don't get scared because Gapit and other software makes these calculations for us behind the scenes in a quick and efficient manner. So it'll do the calculations for us, and you can focus on the biology. And then here's the RR-BLUP model. The slide was made by Brian. So the GBLUP, as we learned yesterday, is an equivalent model to the RR-BLUP model. Okay, so if the appropriate kinship matrix is used or additive genetic relatedness matrix is used, we can have phenotype vector, our grand mean, our random effects account for familial relatedness, and then our random error vector. And then this is how we estimate the grand mean and our BLUPs for our line effects. And then we can use, for example, the RR-BLUP software created by Professor Endelman at University of Wisconsin, which is what I use if I want to do genomic selection in a pinch. And that is it. Wow, I did go through this so quickly that we're actually 3 minutes ahead of schedule. But let's see, how about this? Why don't maybe I answer a couple of questions and then we take a break? Okay, so people in the, you all who are here, do you want to go first? Okay. Yes.
How does this work if you lose a row or something in your field and you don't have enough samples to estimate your weeks in the first?
Yeah. So what happens if you lose a row? So are you talking about a row that has all of the plots in there as like?
So we have our fields, but the planter messed up, so we lost a few replications of one of the other genotype, but we have the rest of everything.
Yeah, I see. So if the planter messes up or something and you lose like a whole bunch of plots, right? These statistical models that we went over are awesome and they are flexible and they can account for those. Okay, so what I would do is I would proceed with fitting the blue or the BLUP model. I would double check your assumptions that everything goes fine. What I would expect is that the standard error for those particular plots that the planter missed, I would guess that the standard error would probably be a little bit bigger. So it's a little bit more uncertainty, but you can still do your analysis. Yeah. These models we went over, they are pretty robust. They're not guaranteed to be perfect or double your money back or anything, but they're still they are flexible enough to handle stuff or to handle things like that. Oh, I see. Okay, sorry. There was a good question that was asked on the chat. So does the Benjamini-Hochberg procedure give any advantages over Bonferroni and GWAS? Okay, here's the thing. Here's an elephant in the room that I did not talk about. Let's say we're doing a GWAS of, say, like 300,000 markers, right? Are you all familiar with the multiple testing problem? So your probability of making a type 1 error, of at least type 1 type 1 error is much greater than, alpha. So the two, I would say, leading methods are the Bonferroni correction, where you just take your genome-wide alpha divided by the number of tests, and then that's your p-value threshold, versus this awesome thing that I prefer, which is the Benjamini-Hochberg procedure, which controls the false discovery rate, which is similar to genome-wide type 1 error rate. Bottom line is, I like the Benjamini-Hochberg procedure better, okay? It is less conservative than Bonferroni. So that means if you have an association that is on the fence to be declared statistical significance, your Benjamini-Hochberg procedure is going to do a better job, or is probably going to declare it to be statistically significant. And with that said, there's a colleague of mine who's starting to say that they don't like Benjamini-Hochberg. So be aware of that. And then in a worst case scenario, you can always do a permutation procedure, where you do something similar to that bootstrap example I was describing, but except you do it without replacement. You can obtain an empirical, data-driven statistical significance thresholds. Do it that way. Okay. Okay, how about this? I'm going to answer one more question on the chat, and then we can take a break until 11. And I will hang around and answer questions until 11 after I freshen up for a second. Will using machine learning for genomic prediction protect us from accounting for population structure? Okay. No, machine learning would not protect us from accounting for population structure. And I would even argue, as I'm going to show actually in the second set of slides, you do not want to factor out population structure for genomic prediction, because you want to measure the entire genetic merit of an individual, be it the genes that are controlling the trait, but also, population structure. So, yeah, machine learning is awesome, but it's not the solution to all of our problems in the world. It should be used in conjunction with statistical approaches. And on that bombshell, I'm going to step out for a second. I will resume our lectures at 11, which is in 27 minutes from now for those of you not in US Central Time. Okay.
leave my stuff here? I'm gonna leave my stuff here. But you turn everything here. Stuff here.
Yeah, you can see that.
Hmm.
आपको क्वेश्चन भी पूछ सकते हैं इसमें हैं?
Hi.
Oh, sure. Tony, yeah.
It's nice to meet you.
Nice
Thanks
to
for,
meet you.
thanks for hosting this. I really appreciate
Cool.
being able to come here and learn some stuff.
It is my pleasure. I love talking about this stuff and I love teaching it.
Yeah, it's really interesting too. It's like, yeah, it's so interesting to me, like how powerful statistics like can be in these sort of contexts.
Yeah, and it is amazing. Like I think it's like I think this is the best, storyline of statistics that one can come up with. Like, I don't know, just like the applications of it and The recording, okay, and I'm going to share my screen.
Hello, sir.
And let's see, I think this is desktop 2, so I'm going to share that. And if I begin my presentation, play from current slide, are we in business? Okay, you all can see this. For those of you on Zoom, can you tell me if you can see the presentation? I see a yes. And I hear a thing that says no sound. Can you hear me? Can someone type yes if you can hear me? Okay. Welcome back. Yes, 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 yes. We can hear you. Sweet. Okay. So the first hour and a half, of this workshop, what we did was we essentially did maybe a semester and a half. Introduction to statistics and how they're used for linear algebra in genome-wide association studies and genomic selection. We covered a lot of ground in the past hour and a half. So between now and lunch, I'm going to take a little more laid back approach, okay? I'm going to show some specific examples of GWAS and genomic selection in crops, okay? And what I'm going to try to do, no guarantees, is finish this by 12 p.m. so that all of you can go enjoy a nice lunch at the nearby campus restaurants in Champaign-Urbana. And those of you who are in different time zones, have a nice break. And I will answer emails. Okay, anyway. So let's go to this presentation. Still getting used to this screen. Oh, are we there? Okay, good. So we already saw this slide, but let's just get our heads into gear, okay? Yes. It is being recorded. Thank you for the reminder. Yep. I see that little like flashing light, that red circle thing. Oh, great. Yep. Thank you, Anup. Okay, so genome-wide association study. We have a genome-wide set of markers. Oh, yes.
Can you just like, what is that little?
Oh, yeah,
Here's a
yeah, yeah.
very simple-
Yeah, I can see how that can be obnoxious. Let me see. Uh-oh. Oh,
No,
man.
it's-
Yeah. Oh, man. So Zoom is saying, hey, we are Zoom and we need to acknowledge that we're here. And yes. Okay, so yeah, GWAS, right? We want to identify genomic regions associated with a phenotype, right? So the way we do that is through a whole bunch of statistical tests of marker trait associations. So the genome-wide set of markers, we fed our statistical model that we reviewed, right, in the previous set of slides. We want to test the null hypothesis, no association with the SNP and the phenotype. So our x-axis here is all the SNPs on chromosome 5 of maize. Our y-axis is our negative log-based 10 p-value that corresponds to this hypothesis test. And our peak association is within the coding region of ZMVTE4 gene, which is good because we identified a peak association that makes biological sense. So let's say that you wanted to incorporate GWAS results into breeding. So you can use that peak association as a potential target for marker-assisted selection. Now, genomic selection, again, what we want to do is, we fit a model where our y variables are trait and our x variable is a genome-wide set of markers and training material. And we use that to predict breeding values using markers distributed throughout the genome. And it can speed up the breeding cycle, right? We use various frequentist and Bayesian models that are commonly used, and most produce approximately the same prediction accuracies. So why did I go over 2 slides that I went over already? Well, I wanted to refresh our memory because I wanted to talk about the key differences between genomic selection and GWAS, okay? First thing is that their overall objectives differ. The main objective of a GWAS is to find genomic regions associated with a trait, okay? In contrast, in GS, what we want to do is we want to see how good of a job our marker sets can predict breeding values of traits, okay? Also, their statistical models are different. A typical GWAS model will test one marker at a time. Meanwhile, a typical genomic selection model will include all markers in the model at once, okay? So here are the differences between GS and marker-assisted selection. GS, you're using your entire set of genome-wide markers for predictions. And because of that, you can account for both the large effect QTL, associations that are statistically significant, as well as a minor effect QTL that you may not declare to be statistically significant in a GWAS. So you can include everything, right? So when you have complex traits, such as following something similar to the infinitesimal model that was discussed yesterday, the GS model will be ideal for that, okay? In contrast, marker-assisted selection focuses on markers linked to genes of major effect, okay? So what that means is you're only accounting for major effect QTL. So if your trait is controlled by a major effect QTL plus a whole bunch of small effect QTL, you're not accounting for the whole genetic merit of the trait, okay? So marker-assisted selection is ideal, or, not ideal, maybe sufficient for predicting simple traits controlled by one gene or oligogenic traits controlled by a few. Okay, please define major effect in MAS. So that was a question on the chat box. So a SNP that might have a statistically significant association in a GWAS, like something that we've seen two slides ago on that vitamin E example. Where you have several peaks from your GWAS, you can still go ahead and do MAS to identify the candidate gene. Yes, so that's a good question. Okay. Why minor effect is more important? Okay, this will be the last chat question I'll answer, and then I'll move on. Well, sometimes your minor effect QTLs, genes might collectively can control more of the genetic signal than the major gene. Okay, so here are some examples of GWAS for identifying potential targets for marker-assisted selection. So these are all papers that I've been on and all research that I've been on. They're kind of old, but their take-home messages are still important. So we have this example of brown stem rot resistance in soybean. Another example for provitamin A and other carotenoid levels in maize grain. Another example that we'll see for the take-home example, I'll go over this afternoon, vitamin E levels. Okay, so first example is brown stem rot in soybean. Okay, so here are some facts about brown stem rot that my co-authors know that I don't, and I'm just regurgitating these. So brown stem rot is caused by this fungus. And it has, yield loss up to 30% has been reported. And it has been mapped to only 12 sources which have been used to develop resistance cultivars. So the bottom line is, prior to this study, there were 3 genes all next to each other that had been identified. And so if we obtain a more precise location of these loci, and do marker-assisted selection on smaller size loci, we could be able to do more efficient breeding for resistance to brown stem rot. Okay, so the data that we had are, let's see, 4 different association panels, and then note that these are the same. So in this panel over here, N1989, the trait was binary. So we had resistant or susceptible, basically. And so we used a binary phenotype analog of the unified mixed linear model, okay? And then the other traits were quantitative, so we used the unified mixed linear model. And stepwise model selection and multi-locus mixed model, I will elaborate that a little bit in this example, but throughout the course of this day, we will touch back on the multi-locus option more and more and more, okay? So here is a complicated plot of GWAS results, okay? We're going to see it a lot today. I think somebody before, sorry, I did not come up with this plot, but I used a lot in my research. So let's go over it. Our x-axis is physical base pair position of the genome. So here, this is a particular region on chromosome 16 of soybean, okay? This y-axis is our negative log base 10 p-value, okay? So wherever we see these these are vertical lines. Wherever you see these vertical lines, those are negative log base 10 p-values, okay? So the taller the line is, okay, the stronger the statistical association between that SNP and that trait, okay? And FYI, just another thing, this horizontal line is our Benjamin Hochberg 5% FDR threshold. So all of the lines that are blue are statistically significant. All the ones that are gray that are not statistically significant, okay? These 2 vertical dotted lines, that's the approximate region of this, RBS1 through RBS gene from prior research. And then this over here, if my okay, my cursor is kind of not working very well, but this is the peak association, okay? Now, all of these purple triangles, that is a measure of linkage disequilibrium of the neighboring SNPs relative to this SNP over here with the peak association. Does this make sense? Okay, so what we are seeing is this. So around the vicinity of the peak association, we see a whole bunch of purple triangles that are taller than the rest, right? So this is a region of LD, of markers that are higher than nominal levels of LD with their peak association, okay? But then as we go further out, we see that the purple triangles are rising up to, you know, let's say negatively small levels, okay? Nevertheless, we see this blue line here and this blue line here, right? So we're still getting statistically significant associations that are way out in the boonies, right? From this peak association here, right? So what's the deal? I mean, we have peak associations outside of the LD window of this SNP over here. And I mean significant associations. So we wanted to explore this, see what's going on. So what we did was we performed a multi-locus mix model, okay? And I'll be happy to talk about the details afterwards, but basically this is a unified mixed linear model, okay? Where you are accounting for population structure, you're accounting for relatedness in the manner that I've shown already, but we're doing a stepwise model selection procedure to randomly select multiple markers that can be included in the model at once, okay? And the underlying rationale behind this is that we can account for multiple potentially independent signals in one statistical model, and that can help us refine a little better our understanding of what's going on in a region with a whole bunch of peak associated SNPs. So bottom line, we tried out this multi-locus mix model and we identified 2 SNPs in this region over here, okay? And then we reran our GWAS using the mixed linear model, the unified mixed linear model, okay? And we used these 2 peak SNPs, we included them as additional X variables in our model, okay? The underlying rationale is that these 2 SNPs can account for that peak association we're seeing around this X coordinate of 33. And so what we saw happening was that we are no longer seeing these, where's my cursor? Or is that a cursor or is it a mouse? I don't know. But these blue lines are no longer there after we account for those peak SNPs from our multi-locus mix model. So we have found similar findings in the further association panels. So that gives us a high amount of confidence that this particular region over here is accounting for the signals we're seeing, you know, multiple base pairs away. So what this means is that we're basically able to refine the region containing these three putative genes. And this should facilitate efforts for both marker system selection and gene cloning efforts. And it demonstrates the utility of GWAS and soybean. Okay. So I'll tell you what. I'm going to move on to another example, but how about I answer one question from you all, and then I'll answer one question from the chat. So does anybody have any questions here? Adam, I'm sorry, not Adam.
Andre.
Andre.
So the Manhattan plot shows as on the y-axis the negative log based stand of the p-value.
Yeah.
What do we use first? What do we use that transformation? And second, does it tell us anything about how strong is the relationship, or just basically how unlikely is find a big just by chance?
Okay.
There's nothing wrong. How strong is the relationship?
Okay, so the question is pertaining to this plot over here.
Yes.
And then the first part of that question is, why are we using the negative log base 10 p-value? Okay, so we're using the negative log base 10 p-value. So like, first of all, what I would use in a statistical test is the raw p-value, right? Because, you know, that's how you do hypothesis tests. smaller p-values are a greater indication of statistical significance than larger p-values. So I argue that it's really hard to visually discern smaller p-values than larger p-values. So the beauty of the negative log base 10 p-value is we're flipping it on its head, right? So smaller p-values make larger log base 10 p-values, and so it makes it more visually easier to see where the peak associations are. And then what was the second part of your question?
So negative p-value, does it tell us about how likely it is? I mean, how unlikely it is to find a value
Mhm.
as extreme or more extreme than that one? But it tells us nothing about I mean, how strong the relationship between that PTL, right, and the position.
Yeah. So, okay. So awesome. So you know the definition of a p-value, the probability of observing another test statistic that is as or more extreme you observe p-value, right? So this p-value and what's being plotted here, the transformation of this p-value, that is measuring the degree of statistical association between your marker and your trait. So are you going with like, this is not telling us anything about the causal mutation, right? And is that where you're kind of going with that question?
I was just thinking that maybe sometimes we're used to think about p-values as correlations, but there's nothing else in the p-value because the association is significant or not. It basically doesn't tell us much about how strong the relationship is. Am I right about that?
So the p-value is giving us amount of evidence against the null hypothesis. So I guess, I can be talked into a lot of stuff. So I think that I can be convinced if I'm not thinking about it too carefully that a lower p-value, stronger evidence of an association. But you can also do other metrics. You can also do like an R-square metric to see the contribution of that SNP to overall trait variability. You can also estimate allelic effect sizes. So if one is not comfortable with resting their case, if you will, on just a p-value alone, there are definitely other metrics that can be used to bolster or detract the evidence of your p-value. Okay, so here's the first question I see here. Okay, by using different models, I mean single or mixed linear model, if we don't get a common SNPs, what would be the best way to? Okay. Let me see. Okay, I'm going to answer this question from 11:21 a.m. Yeah, in practice, you can use multiple GWAS models, and I don't want to get too far into the weeds with answering this question, but basically, it is an acceptable practice to try out multiple GWAS models and to get a consensus opinion about peak associations. I think that's totally fine, totally acceptable, and I support it. Okay, so now let's talk about our second example. And so I gotta tell you, I really like biofortification from its potential to benefit a problem that faces humanity, but also I like it from a quantitative genetics perspective, because the traits that you look at, or at least the ones that I looked at in the past when I was a postdoc, they tend to be oligogenic and they tend to be simpler. So that means that most of the time people think GWAS fails because you hardly ever find anything that's statistically significant. With biofortification traits such as carotenoids and tocopherols, you always find a peak association, or not always, because I'm going to get quoted on that and they won't find anything, but you typically find statistically significant associations that make biological sense. So it's the next best thing after simulations. So biofortification, we want to identify target genes associated with nutrients, okay? And then by finding these genes, one can do marker-assisted selection on these target genes and increase bioavailability of essential nutrients in local germplasm. So vitamin A deficiency, this might be a somewhat old picture, but it affects 17 to 30% of children under 5. Up to 500,000 children become blind every year, and it results in morbidity and mortality. Now, maize is a primary food source in many of vitamin A deficient regions. In particular, the oranger and redder, the more prevalent vitamin A deficiency is. And in a lot of these regions, maize is eaten. So, biofortification is to breed locally adapted maize germplasm so that you have higher provitamin A levels. So I've done work in the past for biofortification. So candidate genes have identified potential targets for marker-assisted selection. And there have even been high provitamin A lines that have been developed in CIMMYT and other organizations called Harvest Plus. And since I've participated in this work, I've heard that a lot of provitamin A maize has become available, and that's just totally awesome. And so previous work, we identified major QTL associated with kernel color that is shown in the first set of slides. And these happen to be near major carotenoid biosynthetic pathway genes. We've also identified evidence for pleiotropy. So in this work back in 2014, what we did was we conducted a GWAS to identify new candidate genes, okay? So that's what I'm going to go over now. A little later on this afternoon, I'm going to talk about determining a minimal marker set to accurately predict carotenoid levels. So a data set that we'll be looking at a lot today, and thus we'll see this slide a couple of times. We have the Goodman diversity panel, which consists of 281 maize lines. They were grown at Purdue in 2009 and 2010. And then our collaborators measured carotenoids for 252 of the lines, okay? Now, just to go into the weeds a little bit so that these results will make sense, let's see, how many of you work in maize? Kind of, sort of? Okay, so I'll tell you a thing that I am not an expert on, but that my colleagues who worked on this work. Maize lines with white kernels do not produce, a non-negligible amount of carotenoids, okay? So in fact, you almost have a near Mendelian association with the PSY1 gene and kernel color, okay? So what we did to factor out the effect of that PSY1 gene, okay, was we looked at only a subset of 201 lines that range from light yellow to dark orange kernel color. So we could go laser focused into the carotenoid biosynthetic pathway and look for genes there in this GWAS. So here is a picture of the carotenoid biosynthetic pathway, okay? The compounds that we measured in our diversity panel, the resulting Y variables that we turned into, I think we use blues here, are colored in red, okay? The enzymes produced by the genes in the carotenoid biosynthetic pathway are in between each of the names of the compounds. And so peak GWAS associations that were statistically significant at a 5% Benjamini-Hochberg false discovery rate are in the man, I'm making this sound much more clumsy than I have to. We found peak associations in the vicinity of the genes that are circled, okay? That is the bullet point, and I know those are really enzymes, so I said that wrong, but we have identified these genes, okay? At a genome-wide level, okay? So a point that we've already touched on is that adjusting for multiple testing can be very conservative at a genome-wide level, especially if you're doing 300,000 tests at once, only the most statistically significant stuff is going to be, only the biggest signals are going to be statistically significant. So what we did was this, okay? Here is one potential idea to overcome the conservativeness of GWAS, okay? Instead of doing multiple testing correction on a genome-wide basis, you can do multiple testing correction over specific subsets of the genome, okay? It's not as, you are no longer controlling for the genome-wide multiple testing problem, and you need to make that clear in your materials and methods when you write it up. But you can, but this is a way to get statistically significant associations if you relax the multiple testing problem a little bit. So what am I trying to say? For this particular analysis, the carotenoid pathway has been studied prior to the study, okay? And these prior works have identified 58 candidate genes, okay? So what we did is we retook the statistical analyses that we already conducted in our GWAS, and then what we did was we only considered markers that were in the vicinity of these 58 a priori candidate genes, okay? And we readjusted our multiple testing problem for there. And because of that, we were able to identify these additional genes that were circled in black, okay? So take-home message is that, we were able to identify large effect genes and maybe more moderate size effect genes as statistically significant. And we are able to identify more potential targets for marker system selection. So this bullet point here, I'm foreshadowing the stuff that we're going to talk about later on this afternoon. So just to get you all excited and thinking about what we're doing this afternoon, are selecting for these target loci sufficient for improving provitamin A content in maize grain? We will answer that question this afternoon, so stay tuned. Are there any questions? Yes.
Yeah, if I can, so you just run the GWAS first, and then you just subset the genomic, like the marker LG
Yes.
in the genomic region that you are analyzing based on your high priority genes.
Yes.
And then how the result is different if you do all the gene associations. So you just able to find SNP that was not coming up with the first analysis.
Yes.
And why is that? So is it because there is less? So I was thinking that the p-values, if you run both and should be similar or?
Yeah. Okay. That is a fantastic question. And I'm going to repeat it so that the folks on Zoom can hear as well. Yeah. So in this pathway analysis, just to go over the procedure. So we started off doing a GWAS across the entire genome. Okay. And then what we did for the pathway-based analysis was we had these 58 genes that we knew about from prior studies. And we then reconsidered only the SNPs that are near these 58 genes. And we redid the multiple testing problem adjustment. And so a result of that, and I think that you said this, so I'm going to repeat it, and if I'm incorrect with what you said, please correct me. So we still have the same models fitted to all those SNPs, the same p-values, right? But the thing that is different is that, okay, I had the exact number of markers near these 58 genes later on in this lecture. If I remember correctly, I think it was maybe like 8,000 SNPs are in the vicinity of these 58 candidate genes. I hope I'm not wrong. We'll see. So imagine situation A, a GWAS, where you're correcting for 300,000 tests, versus situation B, you are correcting for 8,000 tests. you are going to have a much less conservative adjustment for multiple testing in the latter pathway analysis. Great. Okay, there's one question I'll answer here. Okay, so this is saying, hi, Alex, have you tested the genomic prediction using SNPs in the vicinity of these 58 genes? Yes, we have. Okay, so Vivek, thank you for this question and good to see you. So we're going to talk about this particular analysis that you're bringing up. We did try this out for genomic selection, and we're going to talk about it this afternoon. Okay, and we're doing great on time. Let's see. Okay, so I will answer one more question from the chat box and then I'll move on, okay? For a single locus with more than 2 alleles, how would you run a GWAS for a single trait? Okay, so this question is pertaining to a haplotype-based analysis. So are you all familiar with haplotypes? Yeah, okay. So essentially, you can fit the unified mix in your model, and instead of having your X variable being the additive quantification of a SNP, instead you can have like the combined haplotype of multiple SNPs. So that is totally doable, and there is software out there. My former postdoc looked into it more than I have. My biggest concern was, going into the problem is figuring out what constitutes a haplotype and, deriving that in a computationally efficient manner. But I understand that that's not as big of a deal as it is. But I think that doing a haplotype-based GWAS is a fantastic idea. Okay, so my next thing I want to talk about. So here is a software package, okay? Sorry, I'm going to brag a little bit, but this was co-developed by my former postdoc I was just talking about, Dr. Samuel Fernandez, and he just came back into the country yesterday, and he's going to become an assistant professor at University of Arkansas. Like, I think tomorrow is his first day. So, I should update this. Professor Samuel Fernandez and I developed this R package called Simple Phenotypes. And here's why I'm talking about this. The beauty about simulations is that you will know what the actual causal mutations are, right? For simulated traits. So you'll have a ground truth of what's actually underlying your simulated traits. And it's a really powerful approach to see how good of a job a given statistical approach is actually doing with finding true and false positives. And that is the reason why I have this here. So multivariate quantitative genetics approaches have great potential. So when I say multivariate, I mean multi-trait approaches. They can help you identify pleiotropic loci. They can help you improve prediction accuracy, okay? So simulating traits that are correlated. So with this R package, Simple Phenotypes, you can actually specify the correlation between traits, okay? So simulating correlated traits will help you assess the utility because you know the ground truth, you know the genetic architectures of simulated traits, and so therefore you can directly assess your true and false positive identifications. So check out this Simple Phenotypes R package because it can help you simulate univariate and multivariate traits. And the awesome thing is you can use user inputted marker data. So if you have, a data set of genotypes that you're growing out in the field, you can read it into Simple Phenotypes and simulate traits. And a really cool thing that Professor Fernandez incorporated into it is that the output files are directly already formatted for using like GapEdge or Gemma or genomic selection programs. So it does a lot of heavy lifting for you. So with this Simple Phenotypes package, I'm kind of not going to give the punchline to this, but we were interested in how does GWAS differentiate between these two different scenarios I'm going to visualize? So let's say we have pleiotropy, all right? So here's our genomic position, and we have this causal locus, okay? So if this is a pleiotropic locus, it's going to have a direct impact on trait 1 and trait 2, okay? So the other scenario we were interested in is we have two distinctively different causal loci that are right next to each other in the genome, okay? Causal locus 1 is causing trait 1, and causal locus is controlling trait 2. So what we wanted to do is we wanted to see how well can current state-of-the-art multivariate GWAS models differentiate between these stages, between these two scenarios, okay? And when I say multivariate GWAS models, I mean the unified mixed linear model that's adapted for multivariate traits, which I'll show in a second, okay? So, you know, the punchline is that we decided that multivariate and univariate GWAS models should be used in conjunction to come up with a collective bit of insight. And if you're interested in that, check out our Frontiers in Genetics paper. But here is a unified mixed linear model. I'm going over the details here kind of quickly because I think this is the third time I've shown this, maybe the fourth, right? So we're accounting for population structure, we're accounting for familial relatedness. This line random effect is proportional. Its variance is proportional to our kinship or additive genetic relatedness matrix. Okay, so in a univariate GWAS, your Y variable is going to consist of one trait. Okay, in a multivariate GWAS, our Y variable is going to consist of two or more traits. Okay, so okay, we're doing great on time. I'm about to do two genomic selection examples, but are there any questions? Yeah.
Maybe you'll answer this soon, but in multivariate, do you have a single value for your Y variable? Or are you having two distinct Y inputs?
Yeah. So for your multivariate GWAS model here, you are going to have a distinctive input for each trait. Okay. And there's different ways that you can read it into software, but you know, if you want to think about it from a statistical perspective, you know, this Y variable over here, this is a vector, right? And it's going to consist, say you're looking at two traits, and it's going to consist of, you know, so yeah, it's going to be a two-dimensional vector with the two traits, and therefore you need the phenotypic information for both traits. Okay. Okay, so let's see. There's one trait, or I almost call this comment down here a trait. So for a multivariate model, does it matter if the traits are independent or dependent? Okay, for a multivariate model to work, the thing that makes a multivariate model more advantageous than a univariate model for most scenarios is that you have a correlation, a non-zero correlation between your traits, okay? If your traits have a correlation of 0, then there's no advantage to using a multivariate approach. And the rationale behind that is that statistical multi-trait approaches, in their calculations of like standard errors and all that stuff, they are taking this correlation into account, okay? This correlation is going to enable you to get smaller standard errors and hence more precise estimates, and that can translate to more accurate predictions for low heritable traits, basically. All right. So let me, where'd my cursor? Oh, here we go. Okay, so great. So I'm going to show two genomic selection examples, okay? One is an old example, and it demonstrates how GS works, but I am also going to highlight how I changed my mind on something since there, okay? I'm going to show you why to the second example. So this was some work that I've done back in 2014. And this was looking at genomic selection in a biofuel perennial grass called switchgrass, okay? And so the underlying rationale here was that if we use genomic selection on simple to measure traits approximating biomass, we could substantially speed up switchgrass breeding efforts, okay? So let's see. So we evaluated the potential of genomic selection using genotypic and phenotypic resources. So what we had here to look at was a switchgrass association panel. And here is literally a picture of this association panel, okay? So you see how all of the plants look different, like they're like different colors and like some look taller than the others? This is truly a diversity panel. It is a random collection of diversity of switchgrass, right? It was grown in Ithaca, New York from 2009 to 2011. And so switchgrass can be tetraploids or they can be octoploids. So that was the cool thing working with switchgrass. It was polyploid, although it was a polyploid that behaves like a diploid. So we couldn't do anything cool like you can with like potato. And I don't know, is a beet a polyploid? Okay. Not that I'm bashing diploids or anything. And hey, wheat is cool too, but it's an an allopolyploid. So it's essentially 3 diploid genomes, right? Okay. Anyway, okay. I'm over on that tangent, but so it had, and my understanding is that switchgrass, you have northern adapted and southern adapted germplasm. This collection was predominantly northern adapted upland germplasm. So we looked at 7 morphological traits. And me as a statistician, the way I think about morphological is you actually went out to the field and you actually measured them, okay? And then there are also 13 biomass quality traits that we got from this co-author or from this collaborator of a collaborator who published this stuff, who measured it and published it in this paper over here. And then we had about 17,000 SNPs. And we anchored them to a reference genome that was state-of-the-art back in 2014. And we use this to impute missing SNP values, okay? So let's see. We use 3 popular genomic selection models, right? So we use the RR-BLUP model, we use the elastic net, and we use the lasso, okay? So I remember yesterday, Professor Witkowski was talking about if you want to compare various genomic selection models, make sure the models are different, right? So these 3 models are theoretically different from each other. Well, their math says they're different, but their biological rationale differ as well, okay? So RR-BLUP, the way that it penalizes, theoretically, it should perform the best for complex traits, because if you got a bunch of SNPs that are in strong LD with each other, essentially what they're going to do is average out the effect of those SNPs across all of them, okay? And then in contrast, lasso should perform better for simpler or less complex traits, right? So if you have that similar group of SNPs that are in LD with each other, what lasso will do is assign the effect estimate to one of those SNPs and then essentially zero out the estimate of the remaining SNPs, okay? And then elastic net, okay? That is a weighted average of the penalty used for RR-BLUP and lasso. So elastic net is somewhere in between RR-BLUP and lasso. So I taught you all about k-fold cross-validation. We use 10-fold cross-validation to evaluate performance, okay? So our main finding is GS appears to work. So our 3 GS models produce similar prediction accuracies. That's great, right? So high prediction accuracies are obtained for most traits. A morphological trait called standability, which I think is similar to lodging. It had the highest prediction accuracy. So mean prediction accuracy across 10 folds is 0.52. And then the morphological traits tended to have higher prediction accuracies than others. Okay. Okay. People are saying, okay. Anyway, so I am going to preempt a question that I've been asked before, and when I presented this slide, I said the prediction accuracies are high and standardability is the highest with 0.52. I can see some detractors saying, gee, Alex, prediction accuracy of 0.52, that is awful. Why are you saying that's good? Well, there was a paper from 2010 that I always quote, okay, or I always cite whenever I talk about this. Genomic selection, the advantage of that is that you can achieve multiple cycles of selection per unit of time relative to phenotypic selection. So what this paper showed back in 2010 for winter wheat and maize was you can have prediction accuracies as low as 0.2 or 0.3 and still have genomic selection outperform more persistent selection. So if we're getting a prediction accuracy of 0.52, I'm not going to try to tell you that that is the best thing since sliced bread, okay? But it's nothing to frown at or like, dismiss. Okay. Okay. So, now I'm going to show you full disclosure. Something that that was done in the past, and I changed my mind about it. Okay. So to motivate this is the switchgrass example, okay? Our x-axis is principal component 1 of our SNPs, so that's the greatest source of variability. And then our y-axis is our second principal component of our principal component analysis. Oh, there we go, of our 16,669 SNPs. Now, what I did was I changed the symbol and the color of our different 515 accessions of switchgrass by upland versus lowland and by their ploidy, right? Now, I don't know about you, but I'm seeing, at least 3 distinctively different clusters. Do you see that as well? So clearly, there is subpopulation structure going on in this data set, right? And our markers are doing a really good job of accounting for that population structure. Okay, so here's what we did in the past that I no longer agree with, okay? We said we don't care about population structure and genomic selection. We only care about how well are the actual genes controlling the traits making predictions. So what we did was we essentially factored out those 2 principal components to kind of like put the remaining genetic signal on a level playing field and to see how well our remaining genetic signal made predictions, okay? And we did that by taking, we fitted a model with 2 PCs, we took the residuals and did genomic selection on the residuals, okay? I no longer agree with this approach, okay? So my suggestion is don't do what we did in this paper, but instead, so what we probably did is we probably underestimated the prediction accuracy of these traits, okay? So here's the deal. Now, population structure, even though it's a nuisance set of genetic variability, it is still a set of genetic variability. The purpose of genomic selection is to quantify the genetic merit, to estimate the breeding values of an individual, whether it comes from additive, I would say dominance epistatic, and or population structure, right? So here's what we did instead, okay? So here in Illinois, we are really interested in this other perennial grass species or grass genus called Miscanthus. How many of you have heard of Miscanthus before? Okay, cool. Okay, so actually, I think that there's a collaborator in Colorado State who is looking at Miscanthus because we have one of their locations in our data. So Miscanthus is a perennial grass from Eastern Asia. And there are two species that we're interested in, Miscanthus sinensis and Miscanthus cycloploris. And this is actually ornamental too. Like we have some of those in our backyard. Anyway, so where am I going with this? Okay. For biofuel purposes, you use a Northa species. So you essentially cross a cultivar of species 1 with a cultivar of species 2, and you use it for biofuel purposes. And you can get really tall plants, like 12 or 13 feet tall, and get, you know, a lot of biomass, right? There's a problem. Most of the clones of this interspecific cross is just only one genotype, right? So if you have a disease or something coming into North America, it's totally going to wipe out your Miscanthus biofuel crop, right? So there's an urgent need to increase amount of genetic diversity into Miscanthus. So also, so that kind of motivated the study I'm about to show you, okay? So both species have substantial subpopulation structure, as can be seen in these dendrograms, okay? And so the purpose of GS is to quantify total genetic merit, whether it is from the genes underlying the trait or from other genetic differences, as I already said. So let's see. My first postdoc was Dr. Marcus Olatoye. He actually got his PhD from Professor Moore. So you all know Marcus? Okay, awesome. So you know, me and the Morris lab is funny because one of his PhD students did a postdoc with me, and now one of my PhD students is now doing a postdoc with Jeff. So anyway, this is his work. So all these results was created by Dr. Olatoye. So we had a diversity panel in MSI and a diversity panel of second species MSA. So what we did was we fitted a standard GS model, and then we also fitted a PCs only model that only accounted for population structure, okay? And then we compared our prediction accuracies, all right? So if population structure is important, okay, what I would expect to see is this. I would expect to see the prediction accuracies of our standard genomic selection model to be in the ballpark of the prediction accuracies from a principal components only model. Does this make sense? Yes? Okay, I'll say it one more time. Let's say that we fit a model like our standard GS model, right? So that's like our blunt model that I showed you, right? So that is just accounting for all the genetic signals throughout the genome, everything including the kitchen sink, right? So we'll look at that model. Let's compare that model to another model that is tailor fitted to account for only population structure, okay? If population structure is going, has a major say in the total genetic merit of the Miscanthus clone, okay? I would expect to see similar prediction accuracies from our tailor-fitted PC model for population structure as I would from a standard run-of-the-mill genomic selection model, okay? So that's what this slide says here. And here's the result created. This graph is created by Dr. Olatoye. So our x-axis is our different traits that were measured in Miscanthus. This is the first data set, Miscanthus sinensis. And then our y-axis is our prediction accuracy. Okay, so I'm going to pick on this trait over here, HD1. These are the results from our model, different subsets for a training set, but these are the results from the model that accounts for population structure, okay? These are the results from the GS model. So I don't know about you, but these look, oops, oh man, I'm trying to toggle back and forth and I'm messing it up, but these looks similar, right? We're seeing similar prediction accuracies from a tailor-fitted model designed to account for population structure versus a model that, accounts for the entire genetic merit. Okay, so what that is telling me is that a lot of the prediction accuracy we're seeing from the GS model could be accounted for from population structure. Okay, so I suggest not factoring out population structure from genomic selection models because you might be underestimating, the genetic potential of your clones. Okay, so time to have lunch. So yes, question.
Just to be clear that, you are recommending, not to factor out PCs for genomic selection, but that is not all true for, GWAS.
Precisely. Yeah, so let me repeat that because this is an important point. For GWAS, you absolutely want to factor out population structure so you can go after and find those genomic regions that have genes that are doing something to your trait. For genomic selection, I think you should not factor out population structure because you want to measure the total genetic merit of your clone for breeding values. Okay, so so all of the questions. So sorry, I'm going to go to, if I can get this cursor to come down here. There is a question. Okay, so for GWAS use PCs. Oh, okay. How do you define the training population of the model to consider the structure? Do you consider okay. So there's a question about population structure in the training set. I would say just don't worry about the subpopulation structure. Don't factor in your GS model. And then if the training set is representative of the validation set, then you'll probably have similar subpopulation structure. And I'm going down to the weeds. So, okay. So those of you who are here, you all know where to go to for lunch, right? So I agree with Anoop. I think the bread company is a solid place. Like they have some really awesome sandwiches. I love their cookies. Okay, and also they are quick enough that you'd be back here at 1. Oh, you don't think they're quick enough? But there's also like a ton of restaurants all around there. Actually, so all right, I'm telling everybody this who's listening on Zoom, but I love spicy food. Okay, there is a Thai place nearby. Do you know about this Thai place? What is, yeah, Basil Thai. Yeah. So I actually had something too spicy there, and I have spicy food all the time. I gotta stay here, and I gotta help answer emails and stuff, but you all have a good lunch. Okay, and I'll be back here at 1 p.m., which is 58 minutes from now, and Professor Diego Harkin will give a tutorial on G by E based genomic selection. Okay, all right, well, have a great lunch. I'll see you all in an hour or so. No, I'm good. Thank you though. Thank you though.
We did the third one, yes.
Okay. Those of you on Zoom, I'm going to turn off my computer, go up into my office, but I'll be happy to answer questions while I'm eating my lunch, okay?